Okay, this topic covers the last part of the sensory system, so vestibular system. Vestibular system is crucial in balance control and also gaze stabilization. In the inner ear, you can find this structure, vestibular apparatus and cochlear. We're going to focus on vestibular apparatus today, and you can see those semicircular canals. There are 3 semicircular canals: anterior, posterior, lateral semicircular canals. Also utricle and saccule. So those 5 structures constitutes a vestibular apparatus. Vestibular apparatus is 2 parts: bony labyrinth and membranous labyrinth. The bony labyrinth is a bony structure that protects the membranous labyrinth. So let's say this is bony labyrinth and that's membranous labyrinth. Inside membranous labyrinth there's a fluid called endolymph. Between membranous labyrinth and bony labyrinth there's also liquid, and that's called perilymph. Peri means around, so peri lymph, and this is continuous with cerebral spinal fluid. Endolymph has different characteristics to cerebral spinal fluid, that it has higher concentration of potassium ions in the fluid. So let's have a look at the sensory organs of each of those 5 vestibular apparatus. Let's start with the semicircular canal. The sensory organs in the semicircular canal is known as crista ampullaris. So this is the sensory organ for semicircular canal. Which contains hair cells and supporting cells that supports the hair cells. So this is the crista ampullaris, and on top of this structure there's a cupula. Cupula is the gelatinous mass that covers the top of the crista ampullaris. So it's a gel-like substance. So where can you find this crista ampullaris? If we go back to the first diagram, there's a swollen parts in each of the semicircular canal, and that's called ampulla of the semicircular duct. So you can find those crista ampullaris in those ampulla. Ampullae for plural. So let's have a look at hair cell. So this is the hair cell diagram of the hair cell. So you can see different types of hair cells, but the common thing is it has finger-like projections on top of its the cell. So these are the finger-like projections, microvilli, called stereocilia. And the tallest one is called kinocilia. So you can see the height of this stereocilia is gradually increasing towards kinocilia, same as these types of hair cell. It's not shown in this diagram, but after this structure of hair cell, you can see here on this diagram, at the end of the hair cell is very similar to terminal axon. So very similar to axon terminal, in that it can release vesicles and synapses with the neuron next to the hair cell, and this neuron is our cranial nerve number 8, vestibular cochlear nerve. So in other words, hair cell directly synapses with the cranial nerve number 8, creating the impulse to that nerve, delivering that information to the CNS. So how does hair cell know the information about balance? So the information of our balance comes mainly from the position of the head relative to the gravity or rotation of the head. So the hair cell, the stereocilia, actually can pivot around its base. So it can rotate either this way or that way, or the head, or the stereocilia in the hair cell. And each of those stereocilia, there's the mechanically gated channels that sits on top of each stereocilia, and on its gate there's a tip link. It's a cytoskeleton that links the one side of the gate to the next tallest stereocilia. So when there's a force acting upon this stereocilia, and let's say it's rotating this way now, the tip link becomes taut and literally opens the gate of this channel, ion channel. And this ion channel is our mechanically gated potassium channel. And as I mentioned before, endolymph is rich in potassium ion, which means there's a high concentration outside the stereocilia. So according to the chemical driving force, the ions will influx into the stereocilia or into the hair cell. And because it's positive ion, what will happen is it will cause depolarization. So there's another diagram showing the same mechanisms. So there's channels, mechanically gated channels, and there's a tip link that's connecting the gate to the next adjacent stereocilia, the tallest stereocilia, and so is this case. So when there's the rotation towards kinocilium, so there's towards kinocilium, what will happen is it will cause depolarization because of this influx of these positive ions into the hair cell. You can also look at this situation where the rotation happens in the opposite way. So instead of rotating towards the tallest stereocilia, which is the kinocilium, if the rotation happens this side, this tip link will be loose, so it'll be slack. You can imagine it's getting closer to each other, so it'll be slack, so the gate will be closed, which means there'll be no depolarization. So it's in other words, whenever there's a rotation towards the kinocilium of the stereocilia, there will be dipole. The other way around will cause no dipole. So that's how the hair cell detects the direction of the movement. When the depolarization happens, there will be voltage gated calcium channels similar to the axon terminal. Calcium comes influxes to the cell, inside the cell, cytoplasm, and what will happen is it will interact with protein complex and create the exocytosis of the vesicles filled with neurotransmitters, and it will eventually cause the depolarization of the cranial nerve number 8 through ligand gated channels opening and positive ions influx into the cell. So now we know the direction of the stereocilia rotation will cause the depolarization, but what causes the deflection of those stereocilia? It's from the inertial difference between the fluid and the gelatinous mass. So this is the cupula, the gelatinous mass that sits on top of the hair cell. When we rotate our head, the endolymph will lag behind. Because it was initially motionless, and it wants to stay motionless according to the first Newtonian law. So it will tend to stay still, but what rotates is this bony structure and also the gel structure as well. And because that's lagging behind, it will actually create the force in the opposite direction. So if the structure is moving this way, the fluid, because it stays still, will cause the force in the opposite way, and this will deflect the gel, and that movement will create either towards or away from the stereocilia. So let's keep that in mind and apply it to those 3 semicircular canals that we have in our inner ear. So physically, the orientation is shown in this diagram, physical orientation of each semicircular canal. It's 45 degrees away from the mid-sagittal line. The anterior and posterior canals are aligned to that plane. Whereas the horizontal canal sits in the transverse plane, almost horizontal. That's the name, horizontal. You can also realize that anterior canal is in the same plane as the posterior canal on the left side. So this posterior canal on the left, and that's anterior canal on the right. So as the posterior canal on the right and anterior canal on the left. Those 2 canals form a functional pair because they sit in the same plane, but because of the hair cell, the stereocilia direction is the opposite, they can actually detect the opposite rotation. So for example, if the head is moving in this plane, rotating in this plane, posterior canal on the right side will be either maximally rotated. When it's maximally rotated, for example, this anterior on the left side will be minimally activated. So with this design, one can tell which direction the head is rotating by looking at the relative activation of those functional pairs. And because they're all on the 3 different planes, we are able to detect the 3 degrees of freedom rotation of the head. So for example, pitch, yaw and roll of the head rotation. One thing that would be relevant to this semicircular canal activation, so lagging of the fluid, would be when there's no lag in the endolymph. So when the endolymph moves with the endolymph velocity equals the let's say the labyrinth velocity, so membranous labyrinth velocity, there will be no lag, so the cupula will stay stationary. So let's say no deflection in the cupula. And when there's no deflection in the cupula, there's no movement in the stereocilia, thus the mechanical ligated channels will stay closed and there will be no depolarization. So which case caused this equaling the velocity of those two structure? It's when you're rotating a constant velocity. So let's say you are in a merry-go-round. At the start of the merry-go-round, you'll feel the head is moving, but when the merry-go-round rotates in the same velocity for a while, and if you close your eyes, because of this same velocity equaling the velocity of those two, you'll gradually feel no head rotation, and you will feel the head rotating in the opposite way when the merry-go-round stops, and that's where the velocity changes. So whenever there's a velocity change, you'll feel the rotation, but if the velocity stays constant, you will not feel the rotation of the head. In other words, these semicircular canals are particularly sensitive to acceleration of the rotation. So changes in velocity. Whereas the constant velocity, it will not be that much sensitive. Let's have a look at the other two structures of the vestibular apparatus, which are the saccule and utricle. So two organs are shown in this diagram. The sensory organ for the utricle and saccule is called macula. So this is similar to crista ampullaris in the semicircular canal. So macula contains hair cell and supporting cells, as you can see here. And similar to cupula, which is the gel-like mass, the gelatinous mass also sits on top of the macula for utricle and also saccule. Let's just label that as gel-like substance. And the difference between the semicircular canal and this utricle and saccule would be otolith. Oto means ear, lith means stone, so it's ear dust or ear stone. Ear stone, though the little structures are sitting on top of the gel mass, and whenever there's this head movement, the otolith will create different density across the macular area. So some will become more dense, so it will actually deflect the hair cell. Some area will become less dense, so it will make the hair cell stable. So whenever there's a deflection of the hair cell, it will fire the action potential to the cranial nerve number 8. And so this is utricle, and that's saccule. And utricle sits in the horizontal plane, whereas saccule sits in the vertical plane. That's horizontal. So the diagram on the right side shows the plane of the utricle and saccule. So this horizontal plane and vertical plane. So you can appreciate whenever you move forward or side to side, utricle will be activated. When you move side to side, up and down, the saccule will be activated. And the diagram shows the symbolic representation of the arrangement of kinocilium versus the other stereocilia. So the kinocilium was the tallest one, and the stereocilia was the rest. Whenever there's the direction of force acting in this direction, so move towards kinocilium, there will be depolarization. And this diagram shows the dot indicates kinocilium, and the rest of the line shows the alignment of the stereocilia. So for this particular hair cell, if you move this way, this hair cell will be activated, but this hair cell will be deactivated. So by this arrangement, so there's two opposite arrangements similar to the functional pair in the semicircular canal. One can appreciate the direction, whether it's moving this way or that way. And because all the hair cells arranged in 360 degrees, so the angle, actually detecting the whole range of motion, so we can detect the all directions of this planar movement through this arrangement of the hair cell. And this goes the same for the saccule. Okay, I guess that's it. So I'll see you next week.